Hello students, welcome to my channel Biologics 5. I am Niru Sharma and today I am going to discuss about the genetic code. So first, what is genetic code? Genetic code is actually a set of rules which is followed by cells for protein synthesis. It's a set of rules, actually these are codons, and each cell follow these rules to synthesize protein. For my convenience, I divided it into the two parts. First part is interpretation and proposition of genetic code, in which discoveries of different scientists will be discussed and proposition given by different scientists. And in the second part, we will discuss about the salient features of genetic code. So I have divided into two parts and make subsequent videos for that. Today we will going to discuss the interpretation and proposition of genetic code. So in gene expression, as we know, the DNA transcribes itself into the mRNA, and mRNA get translated into the protein, which is a polymer of amino acid, and DNA replicates itself also and make its copy. During both processes, replication as well as transcription, in replication we know the DNA replicates and makes its daughter strand, and that is complementary to their parent strand. And in transcription, the mRNA is formed, which is also the complementary to the template strand. mRNA which is formed is complementary to its template strand. This is mRNA and this is the template strand. In replication, this is a daughter strand. So here we can, in replication as well as in transcription, we can easily predict the sequence of new strand because it is complementary to its parent strand. So it will be complementary to its parent strand, and so the sequence can easily be predicted. As well as in mRNA, it is complementary to template strand, we can easily predict the sequence. But here in the translation, when mRNA get translated into the protein, there is no any complementary between the mRNA and protein. mRNA is actually a nucleic acid and protein is a polymer of amino acid. So the discovery of genetic code is required to predict the sequence of protein, sequence of amino acid protein. So we need to predict the complementary, what's the relation between actually mRNA and protein. There was no any clue about that, but there was several evidences which proved that if there is a change in DNA, or nucleic acid, change in protein also occurs. So change in DNA will result into change in protein. There were different diseases which are caused due to the mutation. So there were several evidences which proved that if there is a change in DNA, addition or deletion of nucleotide occurs, the change in protein also occurs, which is responsible for several diseases. So this criteria actually led the scientists for the discovery of genetic code. Discovery of genetic code is not an easy task, and interpret a genetic code is a quite difficult task. So scientists from different fields are required. Some molecular biologists, some geneticists, some biochemists, some physicists are involved in deciphering and interpretation of genetic code. So the first scientist or physicist, he was George Gamow, who proposed. George Gamow, actually by profession he is a physicist. George Gamow, what he proposed or what he argued? He argued that if these nucleotides, we know that there are 4 nucleotides in DNA: adenine, guanine, thymine and cytosine. If these 4 nucleotides code for amino acids, there is only 4 amino acids, then there will be only 4 amino acids. So he proposed that there must be a combination of amino acid which will code for nucleic acid. Maybe it's a combination of 2 or maybe 3. So he proposed that. That was a very bold proposition at that time. He was that the combination of three nucleotides must be required for coding one amino acid. That was a very bold proposition at that time. Through permutation and combination, we can also predict. As we know, the 4 nucleotides, if they will code for single amino acid, there will be only 4 amino acids get coded. Number of amino acids are 20. The number of amino acids to 20 in biological systems. So if we take
through permutation combination, we can also predict. If we take the combination of 2 nucleotides, there are 4 nucleotides, and if we take the combination of 2 nucleotides like AG, AC, CG, CA, so there can be 16 combination possible. So 16 amino acid will get coded. If we take the combination of 2 nucleotide, then there will be a 16 combination, only 16 amino acid will get coded. If we take the combination of 3 nucleotide, for example, they can be AG, C, CG, A. So there can be possibility of 64 codons, and they can be code for 64 amino acid, and amino acids RNA number is 20. And if we take the combination of 4, that will be a great number. If we take a combination of 4, that will code for actually 256 combination. There will be a 250 combination, and we will code for 256. So here we can see easily that 16 amino acids is less than 20, and 256 is much more larger. 64 is little larger, but there is only this possibility can occur. So George Gamow proposed that the combination of 3 nucleotides must be required for coding one amino acid.
Next scientist in this category is Dr. Hargobind Khurana. Actually, he synthesized a synthetic gene. Dr. Hargobind Khurana. He was Indian American scientist, and he synthesized first gene. Hargobind Khurana. So Dr. Hargobind Khurana synthesized the first gene. Synthesized, we can say RNA polymer. He synthesized RNA polymer. With defined sequence. Then can be homopolymer or copolymer. Synthesized RNA polymer with defined sequences. That play a key role in deciphering the code. Defined sequences of copolymer, of homopolymer and copolymer. Homopolymer we can say, and copolymer. They can be a combination of nucleotide, or they can be a same nucleotide on the sequence. So homopolymer and copolymer, defined sequence of. So he synthesized, he got credit for the synthesizing gene. That's very important for the deciphering the code. The next scientist in the category is Marshall Nierenberg. Marshall Nierenberg and Hargobind Khurana and one more scientist, Holle, got the Nobel in 1968 for their discovery. Marshall Nierenberg. Marshall Nierenberg actually developed the cell-free system. He developed cell-free system for protein synthesis. As we know, the protein synthesis occurs in biological systems on cells, he developed actually cell-free system for protein synthesis in laboratories. For protein synthesis, there is no cell machinery is required in protein synthesis. He developed a system in which he can synthesize protein without the help of cell. So Marshall Lindbergh developed a cell-free system, and even he used the poly-U-mRNA. Actually, gene has been synthesized by Hargobind Khurana. So easily the mRNA can be formed of poly-U sequence. Poly-U sequence mRNA is used by Nirenberg for protein synthesis, and he observed that during protein synthesis a protein is formed which is made up of only one amino acid, that is phenylalanine. The protein is formed and which have the unit of phenylalanine. So he discovered that by using poly-mRNA, only one protein is formed with only one amino acid on them, that is phenylalanine. So from this he discovered, or he proved, that triplet of nucleotide, or we call that codon, we code for single amino acids. He proved through his experiment. Actually, he proved that the triplet of or combination of 3 nucleotide, or the homopolymer, which is 3 number, combination of 3 nucleotide or codon will code for single amino acid. Actually, he cracked the code, and this was quite important discovery, and he through his experiment he proved it also. Next important discovery in this field is of Sevro-Oko enzyme. Sevro-Oko enzyme. It was very important discovery also. Sevro-Oko enzyme, also known as polynucleotide phosphorylase. Polynucleotide phosphorylase. Actually, this is the enzyme used for the synthesis of RNA. This is the enzyme which is used for the synthesis of RNA in a template-free manner. In the biological systems, we need a template for synthesizing mRNA through the process of transcription. Through this enzyme, several enzyme scientists are able to utilize it and can form the RNA without the template. So this was a quite important discovery discovered in the Sevro-Oko laboratory, Sevro-Oko enzyme. Important discovery. So these are all about the interpretation and proposition of genetic code. Through all this discovery and proposition, finally a checkerboard for genetic code has been developed. This is the checkerboard for genetic code, and there are 64 codons in them, and these codons codes for single amino acid. So in my next video I will discuss about the salient features of genetic code. This is all about the interpretation and proposition of genetic code, and these two scientists, Dr. Hargobind Khurana, Marshall Lindbergh, plus one more scientist, Hawley, they get the Nobel in 1968. For their interpretation of genetic code in the field of physiology and medicine. These two scientists, one Hawley, 3 of them got the Nobel in 1968 for their discovery of genetic code, or proposition of genetic code. So that's all for today. In my next video I will discuss about the salient features of genetic code. If you like this video, please subscribe my channel and ring the bell if you want to get notified first whenever I post my next video. Thank you. Have a nice day.